Alright, we're on to the next subject in chapter 3, and this is the idea of a cell cycle. Just like humans and plants and just about every living thing goes through a life cycle, your cells go through a life cycle as well. The majority of the cell's life is spent in a phase known as interphase. Interphase can be subdivided into growth phase 1, or G1, S phase, which is when DNA replication takes place, and then the second growth phase, or G2. Notice during the G1 and G2 phases, protein synthesis is occurring. That's what we just talked about, the making of special proteins or enzymes for the cell. Once the cell has grown enough after the G2 phase, it will begin to get too big to support itself, so it's going to prepare for cell division. And it does this in the process of mitosis. Mitosis can be subdivided into 4 main steps: prophase, metaphase, anaphase, and telophase, or PMAT. If you remember them that way, that might help you. We're going to get into the specifics of all of these phases. So let's talk about the different types of cell division. The majority of your body cells, or your somatic cells, are going to undergo mitosis. So your skin cells, your liver cells, your muscle cells, your bone cells, when they grow and divide, they're using the process of mitosis. The cells that are created in this process are called diploid cells or diploid cells. They have 2 copies of every chromosome. Human cells all have 23 pairs of chromosomes for a total of 46 chromosomes. The process of meiosis is a specialized type of cell division that only occurs in our gametes. This only occurs to form our sex cells, the eggs and sperm, which unite to form a new living being, are made in the process of meiosis. Mitosis is all of the other body cells. So meiosis, or meiosis as it's sometimes pronounced, is gamete cell division. Instead of having diploid cells where you have 2 copies of every chromosome, the gametes only have half the number of total chromosomes. They're unpaired chromosomes, which makes sense because when sperm joins with egg, it forms the 23 pairs of chromosomes to make a new living being. If we look at the overall parts of the cell cycle, this is a picture of a cell in interphase. This is when the cell's carrying out normal functions. It's making proteins, it's growing, it's replicating its DNA. If you were to look at one of these cells under the microscope, it looks like a fried egg, the nucleus in the center, no distinguishable chromosomes. As the cell prepares for cell division, the DNA is going to condense to form chromosomes. Remember that there's 23 pairs of chromosomes. This cell is only showing you 4 different chromosomes, just for simplicity's sake. You can see some centromeres preparing for cell division. Remember, they secrete the spindle fibers. Notice what they're doing in these steps. In this phase, which is called prophase, our nuclear membrane is beginning to dissolve, and you can see these holes enlarging over time. This would be late prophase, and you can see those chromosomes, nice and condensed DNA, and those mitotic spindle fibers, those centrioles, going to opposite sides of the cell. That's what's occurring during prophase. During metaphase is when all of our chromosomes line up in the middle. Metaphase, middle, that's exactly what's happening here. You again see our centrioles at either side of the cell. They're secreting those special microtubules called spindle fibers, which are going to attach to the centromere of every chromosome. And then during anaphase, those centromeres break off. The centrioles kind of act like little cowboys on either side of the cell, lassoing each half of the chromosome to separate sides. And just like what you see here in this diagram of late anaphase, or early telophase rather, you see these chromosome halves getting closer and closer to the center of either side of the cell. Finally, telophase is when a cleavage furrow begins to form. This is when the pinching of the cytoplasm is happening. Notice we get some nuclear membrane reforming. Those spindle fibers are no longer acting as little lassos, and our chromosomes are going to begin to unwind and uncoil to go back to chromatin form. Now we have new cells created. These are now called 2 identical daughter cells. They contain the exact same DNA. They're smaller than the original cell because they haven't had a chance to grow yet. Now they're in interphase, so they'll begin growing at this point. Then they'll go through S phase and then another growth phase, and then they too will undergo mitosis. The steps of meiosis are almost identical. In fact, meiosis is the same process except it's two separate cell divisions. So during the first cell division, you have prophase 1, metaphase 1, anaphase 1, and telophase 1. The big difference between meiosis 1 and mitosis is what happens right here during prophase and metaphase 1. You can see these identical chromosomes are hugging each other. It's a process called crossing over. This is what makes, whoops, sorry my mouse got away from me. This is what makes you different from your brothers and sisters. Otherwise, all of the sperm and all of the eggs created would be identical. This makes every sperm unique and every egg cell unique as well. It's called crossing over, and this is why the products of meiosis are 4 non-identical cells. The second cell division is what takes place after meiosis 2. So if we just take a look at the cells at the end of meiosis 1, these are 2 non-identical cells. The DNA does not replicate before it goes into meiosis 2. The purpose of meiosis 2 is to reduce the number of chromosomes. So if you take a look here in telophase 1, you'll see 2 red chromosomes and 2 blue chromosomes in each cell. At the end of meiosis 2, you only see 1 blue and 1 red. And because of the crossing over that took place in prophase 1 of meiosis 1, these cells, these 4 half cells that are created, are non-identical. They're very different from each other, which again is going to mean you are different from your brothers and sisters, or your children, unless they're identical twins, are different from one another. It's because the process that made the sperm and eggs that formed them was meiosis and created different gametes. This is a chart from your book comparing mitosis and meiosis. You can see the number of cell divisions is different, the types of stages is different. Copying DNA, yes, during interphase, and then it's copying DNA before meiosis 1, but not before meiosis 2, which is why there's that reductive cell division creating those half cells. This video goes through the same process that I just described for you. Again, I strongly encourage you to check some videos out on the internet. Just be wary of which sources you're using. We'll stop here for part 4.